Good evening, all, and welcome to this debate. I am Mary Kufre, and my teammate is Sarah Ajuma. We are the affirmative team for the topic, should drug regulatory agencies require pharmacogenomic testing before approving drugs for public use, or should this remain optional? Pharmacogenomics aims to develop rational means to optimize drug therapy with respect to a patient's genotype to ensure maximum efficacy with minimal adverse effects. Such approaches promise the advent of personalized medicine, in which drugs and drug combinations are optimized for each individual's unique genetic makeup. Drug regulatory agencies should require pharmacogenomic testing before approving drugs to improve patient safety, optimize treatment efficacy, and potentially reduce healthcare costs. By understanding an individual's genetic makeup, tailored treatment plans can be developed, minimizing adverse drug reactions and maximizing the potential for positive outcomes.
Thank you, Mary. Mary and I would like to open our debate by saying, imagine a world where we have the power to redefine what it truly means to have a preventable kind of medicine. We are turning hope into a reality. Pharmacogenomics hold that promise. So by including genetic testing requirements in the drug approval process, we can save lives, moving beyond a reactive medicine into a more proactive care. We can reduce healthcare costs, minimize harm, and ensure medications are both safe and effective for everyone. Mary and I stand firmly in the favor of integrating pharmacogenomic testing into drug approvals to start a new era of personalized and responsible medicine. So we're starting our arguments by stating that many drugs do not work for everyone, and genetics is a major reason why. Take trastuzumab, for example, or Herceptin. It's a highly effective medication for breast cancer, but only in patients who have this kind of breast cancer where there's an overexpression of a gene called HER2, which accounts for only 20% of all breast cancer cases. That's why HER2 testing is now mandatory before starting trastuzumab, and it is mandatory by the FDA. For another example, we have clopidogrel, which is a widely used blood thinner. Before we discovered the connection between a gene encoding for the enzyme that activates clopidogrel in the body and clopidogrel effectiveness, about 30% of patients, depending on their ancestry, were unknowingly prescribed a drug that was not effective for them, leading to a high risk of cardiovascular events like strokes and heart attacks. So by the inclusion of pharmacogenomic testing, we could have prevented these events by identifying those who will have a poor response to either trastuzumab or clopidogrel, helping us match the right patient to the right drug and avoid trial and error prescribing. For our second argument, we're focusing on adverse drug reactions, which are a leading cause of harm, especially in low and middle income countries. For example, take abacavir, which is an HIV medication. If patients carry a certain gene, they can develop severe and potentially fatal hypersensitivity reactions. With genetic testing, we were able to reduce the risk of these hypersensitivity reactions from 5 to 8% to less than 1%. Another example is carbamazepine, which is a drug that is used for numerous neurological disorders, which carry the risks of a very serious skin reaction called Stevens-Johnson syndrome, in Asian populations to be specific. So it was linked to genetics that are prominent in the Asian population, and this link was established in about 2004, while carbamazepine as a medication was discovered in 1960. So there's over 40 years of preventable suffering and wasted money where pharmacogenomic testing could have stopped if we've already identified the patients at risk for these hypersensitivity reactions for abacavir and for these skin reactions for carbamazepine. For the third argument, we'll go ahead for Mary.
The third argument is on reducing long-term healthcare costs. Here, ineffective treatment and adverse drug reactions waste both money and time, and they Examples we have were warfarin and P2C9/4K0RC1 genotyping. Warfarin dosing is notoriously tricky. Patients with certain genetic variants need lower doses to avoid bleeding events. Pharmacogenomic guided dosing reduced hospitalization rates by 30% and was shown to be cost-saving over standard care. Another systematic review found that many pharmacogenomic-guided therapies were cost-effective, even cost-saving, especially in oncology, cardiology, and infectious disease, which means pharmacogenomic testing upfront prevents hospitalization from adverse drug reactions, and is also cost-saving. The fourth argument is on strengthening public trust in new drugs. The public might be hesitant to recommend or accept new drugs due to suspicion on toxicity and adverse drug reaction. But pharmacogenomic data included as pharmaceutical package inserts have made genetic information more accessible to physicians and patients, thus increasing the knowledge on genes and drugs that are recommended for use. This has helped increase public acceptance by enabling informed decision-making regarding drug dosage, selection, and efficacy. Okay, pharmacogenomic testing is costly and resource-intensive. But a recent systematic review by Morris et al. Evaluated 108 cost-effective studies of CPIC-level aging drug pairs. They found that 71% of pharmacogenomic-guided treatments were cost-effective, especially as testing costs continued to drop. Pharmacogenomic testing was also cost-effective in 22 out of 23 studies on clopidogrel and 9 of 11 studies on antidepressants. Even in areas with mixed results like warfarin and HLA testing, the trend is moving towards improved value. This evidence supports the economic viability of pharmacogenomic integration, especially when focused on well-established, high-impact gene-drug interactions. While pharmacogenomic testing has upfront costs, adverse drug reactions cost billions annually in hospitalization and loss of lives. Optional testing risks inconsistent care and greater long-term cost. Long-term cost precision saves more than it spends. Now, also, the field of pharmacogenomics is rapidly evolving and needs more evidence. Science is always evolving, but many pharmacogenomic interactions already meet the highest evidence standards. Including pharmacogenomics in drug approval does not freeze science. It accelerates evidence generation and highlights the need for additional research and validation studies, and also ensures safety and keeps pace with innovation. Healthcare systems must develop infrastructures and workflows, facilitate genetic testing, interpret test results, and integrate genomic information into electronic health records. Educating healthcare providers and patients about the benefits and limitations of pharmacogenomic testing is essential for ensuring its effective implementation. I will now allow my teammates to please continue.
Thank you, Mary. For the last two rebuttals, the first one being limited actionability and evidence outside of oncology. It is true that pharmacogenomics has made the biggest impact when it comes to oncology, but that is just because there is a research funding bias towards oncology rather than anything else. It does not show a lack of potential. But if you've noticed that most of the examples that we've provided in our arguments are outside of the scope of oncology, namely clopidogrel, warfarin, abicavir, and carbamazepine. Pharmacogenomics is actionable, and it's growing fast. Including pharmacogenomic testing in drug approvals will only expand actionability across specialties rather than let it lag. For the last rebuttal is that international regulatory differences. It is also true that different countries regulate pharmacogenomics differently, but that highlights the urgency to act quicker and lead global harmony, as it is a problem that is worth solving. Regulatory agencies already adapt to different population needs with local dosing, contraindications, and drug warnings. Genetics should be no different. We can start with high-risk drugs with high-impact gene-drug pairs and expand from there. Safety should not depend and should not be compromised depending on where you're from or where you live at. For our closing statement, we'd like to say that science is clear and the technology exists, and the human cost of inaction is simply too high. Every preventable adverse drug reaction, every wasted healthcare dollar, and every life lost could be a tragedy we choose to avoid if we act now. Pharmacogenomics is a high-risk, high-reward field, and the risk lies in ignoring it, while the rewards come from saving lives, personalizing care, and transforming how we approve and use drugs. This isn't just about the future of medicine, it is also about doing what's ethical, evidence-based, and long overdue. Let us move forward safer and smarter with the tools that we already have in our hands. These are the references that we've used, and we are more than happy to answer any question that you have.